without further delay, so let's get started. Good morning or good afternoon, everyone. Thanks for joining this session. At the beginning of March, we launched the CosMx SMI data analysis webinar, which cover both the atomic basic data analysis and also some advanced data analysis. Today is the first session of advanced data analysis. I'm Star Zhong, the manager of customer support in China. And in the meantime, my colleague, Jen, the FAS manager, is also in this meeting. And please feel free to put your questions in the chat, and we will answer you as soon as possible. And in this advanced session, we will cover the CosMx SMI RNA data structure and a little bit of the downstream data analysis that using the running the pipeline in atomics and realization out of atomics. So Bruker Spatial Biology provide end-to-end spatial single cell multi-omics solution. So from the sample prep to data analysis. When we talk about data analysis, actually with CosMx SMI data, so data analysis is a multiple step process. So first in our on the SMI instruments, first as the instrument runs, so the instrument will process the raw image data to the image barcode, and then it will also do the initial cell segmentation. So this, we call it primary data analysis, which is on the instruments. And after the primary data analysis, the primary analysis processor data will be pushed to atomics. And in atomics, we will do the secondary data analysis, which is to decoding the image bit barcode to the gene name or tag name. So in this step, we call it target decoding. And then after target decoding, we will assign these transcripts to the segmented cells. And if you found the cell segmentation is not optimal, and you can do the cell resegmentation. After the secondary data analysis, the data matrix will be generated, and then we will move forward with the tertiary or realization data analysis. So all of this secondary and tertiary analysis will be done in atomics. But today, we will talk the workflow. So in summary, the CosMx data analysis can be shown in this chart, that after primary data analysis, and then we do the secondary analysis and tertiary or realization data analysis. So before tertiary data analysis, we need to check the quality, especially the image quality and the cell segmentation quality. If you found the CosMx SMI, if you found the FOV images is not good, and then you might need to remove this data. And if you found the cell segmentation is not optimal, then you can adjust some parameters and do cell resegmentation in atomics to get the optimal cell segmentation. After you found the image is good and cell segmentation is good, then you can move forward with the tertiary data analysis. Tertiary data analysis actually is the realization data analysis, which is to convert your matrix, your CSV matrix or your spreadsheet to the plot. So in this tertiary data analysis, we will cover basically the data structure and the contents of SMI data, which is the major focus of this session. And you can also analyze the data in atomics and do the realization in atomics. So which will be covered by my colleague in the atomics basic sessions. And also you can run the pipeline in atomics and realization the data or make plots outside of atomics. And this session, this part we will also cover in this session. And you can also do the de novo data analysis outside of AtoMx using the open source tools with the exported data. And except this gene expression matrix, we also generate some FOV images. So you can use the open source tool to do the image viewer with the exported data, like the Napari that we have developed a Napari CosMx plugin that can realize the data to generate very beautiful publication-ready images. So in this session, we will cover the major part is the data structure and the contents of SMI dataset. And then we will spend a few minutes to cover this part to run the pipeline in AtoMx and visualization out of AtoMx. So let's go to the first part, the SMI RNA data structure. If you create a study and then in the study page, there will be an export button. So to dig into the data structure, so we need first to export the data. So after you click the export button, there will be multiple options, and then let you to check to export the data. Actually, the data can be partitioned into 3 categories. The first category is the CSV file. Actually, this is the spreadsheet that generated from the secondary analysis. And then the second category is the tertiary analysis object. So this is the processed data that derived from the AtoMx pipeline. So you can see there is a CRAT and TileDB file type. And the third tab is the low data, basically, which is the images, FOV images, or some decoded very low data in this low data folder. So first, you need to export the data by checking these options. And I will use the AtoMx 132, this version, to demonstrate the data structure. So maybe your AtoMx is upgraded to 1.4 and version 1.4, and the data structure will be a little bit different, but generally it is the same. So first, you need to export the data, and then after export, you need to, after export, actually the data is exported to the remote server. So we call it SFTP. So after that, you need to transfer the data to your local machine or local computer. You need to connect with the SFTP with the remote tools. And then you can find the credentials by clicking the show export details, and then you can find the remote server credentials in this page, in this window, and then you type these credentials in the remote tool, and then you can transfer the data to your local computer or to your local machine. So I think any remote tools will work, and you can using the FileZilla, for example, and maybe you can you can also use the WinSCP to transfer the data from the remote server to your local computer. Except these two frequently used remote tools that Broker has also developed a remote tool called CosMx Data Downloader. So it is a terminal based. So you need to type some command and then to download the data from the remote server. So and you can reach out to your local FAS or support to set up the CosMx Data Downloader. So I from my experience, I recommended that you use this tool because it is much faster and you can check the progress of the download, and also you can check the file integrity as well. So if you want to use this tool, please reach out to our colleagues in your region. After the data was transferred to your local machine, then there will be a folder for each export. like here, and under this atomics exported folder. So this folder will with the name of your study and some timestamp. And under this folder, there will be a flat file folder, low file folder, TileDB folder, and a serial object file, the RDS file. So first, let's deep dive in the CSV file. And after you check these 5 CSVs, and then these 5 CSV will located in the flat file. So under this flat file, there will be a subfolder named with the flow cell name. And if you export the data from a study contains multiple flow cells, then each flow cell will be each subfolder will be a flow cell. So under each subfolder, there will be 5 CSV files. So you can see in this screenshot. First, let's talk about the polygon files. So the cell polygon CSV file actually is the cell boundary file, which contains the cell boundary information of the segmented cells. So if you check out, if you look at the data, look at the image in atomics, then you can see the cell segmentation, the cell boundary in the white lines, white polygons. So this actually is the cell polygon information, and then in this CSV file, then this polygon file is a spreadsheet of seven columns describing the cell boundary information. So it has the FOV ID and cell ID within the FOV, and the cell ID within the flow cell, and the cell polygon position, the cell boundary information in the local within the FOV or within the flow cell. So these are all the cell boundary coordinates in local and in global. So you can get all these informations, cell boundary informations in the polygon files. So next is the expression matrix file. So this expression matrix file contains the cell by gene expression matrix at the gene level. That means it has a quantification of each gene, what's the gene counts in the cell. And this is also a spreadsheet with each row representing a cell. So each row representing a cell, and the columns representing the features. And like here, there is FOV ID and cell ID within the FOV. And there also contains targets. So this targets has 3 categories. So the first category is the gene name. So actually, this is our interested targets. Like a panel, if you run 1,000 plex panel, then there will be 1,000 genes. So these are gene names. Is one category. Another category of the target name is the negative controls. So these negative control probes start with negative. So if the number of negative probe depends on the panel that you run. In 100 plex panel, it has 5 negative probe targets. And if you run 1,000 plex, it has 10 negative probe targets. So and the third category of the target is the system control, which we also call force code. So this force code is with a bit code, but no corresponding hybridization mRNA hybridization probe. So the number of system control is also depend on the panel that you run. For 100 packs, it has 10 system control, but for the 1,000, it has, I think, 197. So the number is different, depends on the panel. So these are the this is the content of the expression expression matrix. And the third is the FOV positions. So the FOV position file contains the flow cell level FOV center coordinates. So I take a screenshot from atomics, and then so this CSV file contains the FOV center coordinates. in the pixel and milliliter unit. So like in this screenshot, it has 4 FOV, and it shows this center positions of each FOV. So you can see. And the third is the metadata file. So metadata file contains the cell level information of segmented cells. So this information can be divided into two categories. One is the basic information, like the area, like the diameter, etc. All of these are some basic information, and the other one is the derived information. Like if you run the pipeline in Atomics, and then if you run the cell typing, then it will show you these clusters. Like here, if you run the Leiden clustering, it will show you which cluster this cell belongs to. Like here, it belongs to me cluster 9, 6, etc. So all of these are called derived in metadata. So this spreadsheet has more than 100 columns, depend on how many modules that you run in the Atomics pipeline. So if you check all of these column names in the metadata, so it can be more than 100, and you can check this GitHub blog in our Scratch space. So in this blog, in this poster, we have a very detailed definition of each column name, like FOV, the FOV ID, FOV number, and center, where local pixel, etc. So all of this column name was defined, was described in this blog. And there is also a legacy Excel table. So I think our local FAS have this legacy Excel table, and it also has this metadata column name definition. So you can check this definition as well by reaching out to your local FAS to get this Excel table and to check the column definition of each column name. And the last CSV file is the transcript file. So this transcript file contains the transcript level matrix, contains the transcript coordinates within FOV, or flow cell, or flow cell, or within flow cell. So if you check in Atomics, you can see this dot, right? Each dot represent an transcripts. So then in this transcript CSV file, it has the information of each transcripts. Each row in the spreadsheet represent one transcripts. It has this FOV ID, cell ID, and within FOV, and cell ID within the flow cell, and the transcript position within the FOV, and transcript position within the flow cell. And it also has this where this Z slice where this transcript come from, which Z slice, because in CosMx SMI we will take 8 Z slice for each FOV. So you can, if you want to reconstruct the 3D image, and then you will need this information that to check which these slides that this transcript come from, and also it will show you the target name and cell composite, the cell compartment that the transcript located in. So this is a very comprehensive transcripts metadata. And if there is you need to pay attention that if the cell ID is 0, so like here, you check the cell ID within the FOV. If it is 0, that means this transcript is not assigned to a cell. And then if we translate it to biology meaning, that means under this segmentation settings, this transcripts is extracellular transcripts. That means this transcript is not assigned to any cells. So you need to pay attention to this. Why there is an zero in the cell ID. Next, we will go to the tertiary analysis object. So after we running the pipeline, so the process data, like you run the UMAP, you run the PCA. So this derived data or from the processed module will be saved in the tertiary analysis object. So there is two kind of objects. One is the serial object. Another one is the TileDB. So under the atomics exported folder with the name of the study, so you will find these two folder or files. So let's take a look at the serial object first. So in the export button selection, you can select, there is multiple options. So we will talk about what's the difference between different options. And then you export the data, then you can get this RDS file. This is the serial object. So serial is a very popular single cell genomic analysis tool in the community, and it has also widely users in the community. So this is actually a serial object is a data container in R. It contains multiple data tables which are linked to each other. Like here, there is a different slots, contains the low data, normalized data, scaled data, etc. And it also contains the cell metadata and cell multi-dimensional metadata like the reduction data and regular or normal cell metadata, and it also contains other slots. So you can learn more about the serial object structure and in the wiki page. And also there is also a very good resources to introduce the serial object in this link as well. So there are 4 options to export the serial object. So you must check the export a serial object checkbox in order to export the serial object. So if you only check this export a serial object, it will only contain the cons metadata dimension reduction. It will not contain the transcripts coordinates and cell polygon information. But you can also, if you check export, if you additionally check the export serial contains transcript coordinates, then it will contains the transcript coordinates. And if you additionally check the serial contain polygon coordinates, then it will contain the cell segmentation information. If you check both of them, then it will contain both of these informations. So let me show you the details of the difference if you check different box to export the serial object. First, if you only check the export a serial object, you can see in the image slot. So in the image slot stores the spatial information like the transcript coordinates and cell boundaries. If you only check this box, then in the molecular and boundary slot under the image, then it will be empty. So in the boundary slot, it will only contains the centroid. That means the center of the cell position, not the cell boundary. So it will only contains the position of the cell center, the center of the cell. And if you check the transcript coordinates, then you can see in this slot under the molecules, then there will be transcript information, but in here there will be empty. And if you check the polygon options, then there will be, except the centroid position, it will contains the cell boundary that called segmentation. And if you check both of them, then you will get both informations. So this is the difference when you export the serial object. So you can export, you can check the option on your own, depends on your own needs. And after you export the serial object, you can go to the CIRAT vignette. So you can go to the CIRAT official website and check the vignette and using the CIRAT functions or to do the pre-processing and data analysis or visualization, etc. This is the general or overall the vignette. But if you want to read dedicate CosMx SMI dataset, then you can go to this vignette under the CIRAT website. Then it will show you how to visualize the data and then how to, especially the spatial visualization using the CIRAT function. So here is a very, I will briefly go through the CIRAT structure. So it is slightly different from the regular CIRAT object. So first is the assay. So in the assay, there is a different slot like the low counts data, scale data, etc. So in the atomic exported CIRAT object, each target category will be recognized as an assay. Like here, there is a false code RNA and negative probe. And if you run the normalized data normalization module in the atomics, then it will generate another assay called RNA normalize, and there is a analysis ID, and this assay will be generated from the RNA normalization module. And under each assay, there will be different slot to hold different data types. So you can check the CIRAT wiki for the detailed information. And some of them, because some of them are empty because we do not to apply the functions on the data in atomics, like the counts will be the low counts and the data will be the low counts as well, but the scale is empty. So many of them will be empty, but some of them are not empty and will have some informations. And the metadata will be cell level metadata. So in like I told before that the metadata has two categories. One is the basic information, another one is the derived information. So if you run the module like QC, Latent clustering, cell typing, neighborhood analysis, etc., so this module will generate the information and store this information in the cell metadata, like the RNA NB cluster. So this actually running if you run the Latent clustering or running in situ type, then you will generate the cell type information in the cell metadata, and then this the information can be found in the metadata slot. And also in the ligand receptor and the differentiation gene expression module, you need to select some metadata information, right? And then it can also, this metadata information can also serve as the input for those modules. So next is the active assay. Default is the RNA, and then you can switch to RNA normalized to continue with the data analysis. Others graph like this slot will this module this is the adjacency graph matrix. So this KN graph is generated in actually from the neighborhood network module. So expression space module. So this you can see, if you expand this slot, you will see this information, and neighbors will need generated from the Latent clustering. If you run this module, then the processed the data will be stored in the neighbors slot. A reduction as well. So if you run the PCA module and UMAP module, then you will find the processed data in the reduction slot. Then you can check, like here, this is a UMAP, this is a PCA, and then you can access this processed data by the CIRAT functions. And the image slot contains the spatial information like the molecular, the transcripts coordinates, and the cell segmentation or cell boundaries. The project name default is Surat object if you act in the atomics exported Surat, and the MISC is the user defined information. Here is 0, and if you check the version, then it is from the Surat version 5. And if you Surat package is version 4, then it may be not compatible, so you need to upgrade the Surat to version 5. And the command is the command list or history of code that you use it to analyze the data, but in here it is empty, and the tools is empty as well. So I just briefly go through the structure. If you want to find more, then first you need to know the Surat object structure, and then you can found the processed data in the Surat object. Another type of processed data is the TileDB. Then you can check the TileDB box and export the TileDB. The TileDB object is a collection of the pointers to the RNA counts, normalized RNA counts, negative control probes, and force code. SOMAs. So SOMA is a stack of matches annotated. That means each SOMA is follows the AND shape. So AND shape is actually also a data structure that is very popular in single cell data analysis. If you want to learn more about the AND shape, then you can click this link and then to go to the official website to go through this AND shape. So those files cannot be opened as usual as we do that to just to double click and open this folder. So you cannot touch the file except through the TileDB accessor function. So you can only use the functions to access the data or to pull the data from the TileDB using the TileDB accessor functions. Fortunately, that brokers also generate a very detailed variant on how to pull the processed data from this TileDB, and you can refer to this either of these two link works. So you can open the link and go through the variant to on how to pull the data from the TileDB and then do the realization outside of atomics. So this is a detailed information of where the processed data was stored in the slot. Like here in the OBS slot, it will have this cell metadata, and in the OBSM, it will have the pathway data and dimension reduction data, etc. And you can check this table and check our variant for the detailed information. So the last data type that we exported is the low files. So low files basically is the image file, FOV image files. So under the atomics exported folder, and then you can find the low file folder. And in under the low file export, there is also multiple export options. So you can have 4 options that to or 5 options or 6. I think it is like 5 options to export the low data. If you want to export the low data, you must check the export low data file box, and after that you export. The hierarchical folder structure is like this. And then if you check the additionally check the export morphology 2D folder, and then in the cell stats folder, there will be a morphology 2D folder. And if you check the spot file, additionally check the spot file, then it will have a spot file folder under the low file folder. If you check others, then it will if there is other data, then it will be exported. So in this example, because this data is run very early in our, I think, prototype instrument, so it has the morphology 3D, but I think in the commercial instrument, there will be no such data. So this is just an example for demonstration. And if you check all of them, then it will contains all of the data. So so you can for the details of this folder contents or the data types or what's the content of the data, then you can refer to our manual that in our CosMx SMI data user manual, there will be a part that to talk about the details of the exported data. So you can click the link and then to download the data to read the page 88 to 90 to 92, and then you can find this very detailed information of these data contents. So, and I will not go through the details with you in this session. So usually we recommend do not check these two boxes. Only check the morphology 2D folder to export the raw data, because the spot file is the spot calling file. So it is very big, and it will take a long time to download. But users rarely use this can because they do not know how to decode in this spot file. So users will rarely use this. I think in CosMx 1.4, there will be no such option. And only in the previous version, there will have the export spot file option. And this other data files is also not necessary, because we barely use these files as well in the downstream data analysis. So we can export the 2D morphology image, because we can generate the composite images from this 2D image. And we can also use Napari to import this raw data to generate the images to show the cell type and transcripts spatially. So we recommend that users select only the 2D folder to export the raw files. So until now that we have finished our data structure and contents of SMI dataset. So, and my colleague also covered this run pipeline in CosMx and realization in CosMx. So now next I will take a few minutes to go through this run pipeline in CosMx and realization outside of CosMx. After you know this data structure and contents, then you want to analyze the data. So there is three options. So I mean, so for this tertiary realization data analysis, so if you do not have server, because CosMx SMI data is a very big data, it can have millions of cells and billions of transcripts. So it is a very big data. So sometimes maybe you do not have a very powerful server, and then you don't have the resources to run the de novo data analysis on your local machine. Then you can use CosMx as the server to run the pipeline. So then you can run the pipeline in CosMx and realization outside of CosMx. So first, you can run in the pipeline in CosMx, like you usually do in CosMx. You set up the pipeline, you set up the workflow, and then click run. Then all of the modules will be completed, will be run in CosMx. And then after the pipeline was completed, then you can export the processed data through the serial object or through the TileDB array. These two data type also contains this processed data. And then, as I mentioned, that each module, if you run the module, each output of the module will be saved in the tertiary analysis object. So after export this processed data, then you can pull the processed data from these two from TileDB or Surat, and then you can make your own customized plot outside of Atomics using R or using Python. So here is an example, like you can pull the data and then make the plot. So this is the workflow of running pipeline in Atomics and then visualization outside of Atomics. So after you export the process data, then there is two ways to do visualization outside our Atomics. The first one is following a variant. There is an example, I think, using the liver dataset. So there is an example. So you're following the variant to pull the data, and like here you pull the UMAP process data, and then and also the cell type process data as well, and then to draw the plot outside our Atomics. So this is PCA, this is UMAP, and then this is the spatial plot. The second is that you can also use the Surat, following the Surat package variant, that there's a dedicated dataset, a dedicated tutorial of CosMx SMI dataset, and you can use some of the functions to draw the plot. Like here, then you can using the function apply this function on your exported a Surat object, and then to draw the plot as well. So there are two options for this second data analysis option, that is to run pipeline in Atomics and then to realize outside our Atomics. And for the exported data, each data category has its own usage. So we will talk about more on our next advanced session on how to use the CSV file to load to Surat, and Giotto and SquidPy to analyze the data, CosMx SMI data de novo de novo outside of Atomics. So let's summarize what we talk in this session. So first, we talk about the general overview of the data analysis workflow from the primary analysis to tertiary data analysis. And then in this advanced session, we majorly talk about the data structure. We talk about the CSV file, what's the contents in each CSV file, and we also talk about the processed data, the TileDB and Surat. You can run in the pipeline in Atomics and then export the Surat and the TileDB and do the visualization outside of Atomics. And we also talk about the raw data. We recommend that you export the raw data only by only checking the 2D morphology folder. And except the data structure, we will we also covered this part, like very briefly, that you can run the pipeline in Atomics and do the visualization out of Atomics using the R or Python or other visualization open source tools. And with our next session, we will talk about how we can use the exported file to do the de novo data analysis outside of Atomics using open source tools. So that welcome to join our next sessions, and stay tuned. That's all I want to share. If you have any questions, please put your question in the chat, or if you want to talk, then unmute yourself and then ask questions. I will try my best to answer your questions. Thank you. Oh, wait a second. I also want to share some resources like this are open GitHub, NanoString GitHub is our open source tool that will be hosted in this GitHub site. And as I mentioned in our there will be a variant, right? If you navigate to the export function variant, it is a HTML file, and then if you download it and open it, it will show you how to pull the data from the TileDB and the CRUT as well. So there will be some introduction of the data set, and then on how to read the data into the R or Python, and there will also some serial object data as well. So you can check this variant for the detailed information. And in the nanostring analysis uh, part, there will be very detailed information on how to pull the process data from each module. Like here, if you want to plot the PCA data in your from your TileDB data, then you can use this command or use these scripts to pull data from the TileDB, or you can also found the PCA data in the serial object. So both of this process data works with in this variant. So you can check each module, and then to pull the data and make the customized plot that you want, like Leiden constraint neighborhood, spatial expansion, and all of them, etc. So you can follow in this tutorial, following this variant to do the customized visualization. And another one is the CosMx analysis scratch space. This is our dedicated blog that we put in the GitHub for the CosMx data analysis. If you want to go through the detailed CSV file, then you can check this block. It will show you this metadata column definition, like here FOE position find file. It will show you this definitions of these column names. You can check very detailed information in this block and expansion metrics, etc. So it is very detailed. So thank you all. That's all I want to share. Thank you very much. If you have any questions, welcome to put it in the chat and unmute yourself.